Hello, everyone. I'm going to be covering the first part of Chapter 9, which is Physical and Chemical Controls of Microbes. This first mini lecture is on controlling microorganisms. So, controlling is all about the degree of exposure to potentially harmful microbes, and that's the ones that we mainly look at. We're not so interested in controlling microorganisms that would be not harmful to us. There's a number of different methods of microbial control. Most of these are used outside of the body, so they're used on inanimate objects. That would include sterilization, disinfection, decontamination, and antisepsis. This table, which is from your book, talks about the different concepts in antimicrobial control. So the first of those would be sterilization, and this is a process that destroys or removes all of the viable microorganisms, so everything including viruses. Disinfection is a little bit different. It's a physical process, or it might be a chemical agent that destroys a vegetative pathogen. That means one which is growing currently or metabolizing. But it will not take care of bacterial endospores, and it will remove the harmful products that microorganisms may secrete, things like toxins. The next level then would be decontamination or sanitization, and these are cleaning techniques that will remove microorganisms as well as debris, and it will reduce contamination to safe levels. It does not completely get rid of contamination. And then the last would be antisepsis or degermination, and this will actually be on the body. This is reducing the number of microbes on the human skin. So it's a form of decontamination, but on living tissue. The top three are all on inanimate objects. Down here, we are actually talking about on a body or skin. So what makes microbial forms resistant to antimicrobial agents? And there's a number of things we look at. We mainly want to target, once again, microorganisms that can cause infection to our body or perhaps spoilage of something in the environment. One of the things that is most difficult out of this is bacterial endospores. They're considered the most resistant microbial entity. That endospore is protective against antimicrobial control. The goal of sterilization, remember this is the highest level of antimicrobial control. The goal of it is the destruction of bacterial endospores. In the process of that, it will then obviously kill all less resistant microbial forms. So its ultimate goal is really going after those endospores with the idea that everything else that's less resistant will be killed. This is a really great table in your textbook. It's showing the difference from physical agents, chemical agents, and mechanical removal methods. These are all microbial control methods. And then it's showing you what ends up happening at the level of destruction of the microbes, from disinfection, sterilization, antisepsis or degermination, and decontamination and sanitation. This is a really great one to see how some of these not necessarily overlap, but many of the same agents or methods work for different things. So let's talk about some vocabulary. We need to be able to differentiate between the agents of microbial control and the processes. So the agents are what is actually being used. The process is how those agents actually work. So when we talk about sterilization and disinfection, we're talking about the process of these two. But what we use in those, or the agents we use, would be things like a bactericide. Side always means killing. It's much like homicide. So when you think of bactericide, you should think of killing or destroying bacteria. This would include everything except for the endospore stage. Fungicide is obviously going to be a chemical that kills fungal spores. Virucide is what is going to be known to inactivate viruses, especially on living tissue. Sporicide is something that's capable of destroying bacterial endospores. And then a germicide or microbicide is a chemical agent that kills microorganisms. So it's a much broader term. It could include bacteria, fungi, or perhaps viruses. So continuing with this agent versus processes, sepsis is the growth of microorganisms in the blood of other tissues. So the opposite of that would be asepsis, and that's any practice that would prevent infectious agents into sterile tissues, which could cause infection. So we want to prevent that. That's why we use things like aseptic techniques, keeping it from contaminating or infecting things, and it's practiced in healthcare. And this can range from sterile methods to antisepsis. We use antiseptics, which are the agent, to do antisepsis, which is the process. So an antiseptic would be some sort of chemical agent that can be applied to body surfaces. So it can be applied to either skin or mucous membranes, or perhaps a wound, something that we would use to clean out a wound, and also surgical incisions to prevent any kind of growing pathogen. We often use these, for example, in preparing the skin before a surgical incision, and they use iodine as the antiseptic. With a root canal, they'll use hydrogen peroxide, another great antiseptic. And then we might use ordinary hand washing with a germicidal soap, and that would also be considered antiseptic. So there's also some processes and agents that will keep things at what we would call a standstill, that prevent things perhaps from growing more or growing. And so the term stasis or static mean to stand still. If we have bacteriostatic, this is going to be a chemical agent that prevents the growth of bacteria. And that might be either on living tissue or on an object in the environment. Obviously with fungistatic, that's going to be one that would inhibit fungal growth. And many of the antiseptics and drugs have this microbistatic effect. And it's because we can use that without harming humans, where if we had used a microbicidal compound, it may actually hurt the cells. So we might use these microbistatic agents to keep an area from getting infected or having bacteria grow. And once again, we need to always remember that an agent that ends in "cidal" does not necessarily result in sterilization. And remember, sterilization is that level above just killing growing bacteria or growing microbes. It's where they actually kill the endospores. So a bactericidal agent may not kill the endospores, but would definitely kill any growing bacteria. So when we look at perhaps a clinical setting or a laboratory setting, what are the practical matters that we need to think about in microbial growth? Some that you should consider are, does the application require sterilization, so endospore killing, or is disinfection adequate? That would be depending on the presence of endospores. Is the item to be reused or permanently discarded? These are other things to think about. If it will be reused, can the item withstand heat, pressure, radiation, or chemicals, or will that damage whatever the instrument is? And then thinking about what control methods are appropriate, and will it leave something, a residue or something behind that we don't want? Can the agent penetrate whatever it is that we're applying it to the extent that it needs to? And then is it cost and labor effective, and is it safe? Those are all things that are thought about. So when we're talking about microbial control, we're talking about microbial death to some degree. And death is defined mainly by the permanent loss of reproductive capability. And that means even under the optimum growth conditions, that it cannot grow, it cannot reproduce. And that is how we describe death of a microbe. So when we're looking at the factors that might affect death rate, you have to understand that, first of all, we're not going to be killing the whole population instantaneously. We're looking for a certain threshold once that microbicidal agent is met. And obviously, death would continue in an increasing fashion over time, or the concentration is increased of whatever agent we're using. Active cells tend to die more quickly than less active cells, and this is because many of the agents target a growing cell. And then eventually there is a point at which the survival of any cells is unlikely, and that point is equivalent or is considered sterilization. So other factors that will affect the death rate is how many microbes we have. If there's a large number of microbes, it will take longer to get sterilization. Also, what the nature of the microorganisms is in the population we're trying to control. Generally speaking, there will be a mixture, and some of those may have endospores or may be more difficult to kill. Also, the temperature and pH of the environment, these all affect the death rate. And then lastly, the concentration of either the radiation or the chemical agent, the dose and intensity of that will affect the death rate. The last couple of things that also may affect it is how the agent works. Does it kill or inhibit the microorganism? And how it does that will affect the death rate. And lastly, there may be things that are present that could interfere and inhibit our antimicrobial agents. Sometimes saliva, blood, and even feces can inhibit the action of disinfectants. And they can even inhibit the action of heat. So keeping those things in mind, those will all affect the death rate. So when we look forward, then there are the modes of action of antimicrobial agents, which I'll talk about more in the next lecture of this chapter. Antimicrobials have a ton of different targets that they might be affecting. And the least selective agents, so the agents that work more widely, will be effective against the widest range of microbes. So things like heat and radiation, which are not specific for a particular microbe, will work effectively against a very wide range of microbes. On the other hand, if you have a drug that is only targeting a particular cellular component of perhaps a bacteria, then it will really only work on that. It won't work on the other microbes that might be present. So when we're talking about physical and chemical agents, which I will get into more in my next lecture on Chapter 9, the targets of physical and chemical agents include the cell wall of the organism, the cell membrane, the cellular synthetic processes, so whatever the cell is trying to make things, it will disrupt that. And lastly, proteins. It may be that it denatures proteins that are necessary for the growth of that microbe. This is kind of a broad general overview of control of microbes. We'll talk more about physical and chemical control in the next lecture.